Presence of such airborne nutrients in microbial growth, if not detected, can affect experiments in biochemistry and molecular biology, as well as studies of microorganisms growing in allotropic environments. Natural substances also can directly inhibit microbial growth and reproduction in low-nutrient environments. These agents include phenolics, tannins, ammonia, ethylene, and volatile sulfur compounds. This is a means by which microorganisms avoid expending limited energy reserves until an adequate supply of nutrients becomes available. Such chemicals are also important in plant pathology and may aid in controlling soil-borne microbial diseases. Quorum sensing in microbial populations. For decades, microbiologists tended to think of bacterial populations as collections of individuals growing and behaving independently. More recently, it has become clear that many bacteria can communicate with one another and behave cooperatively. A major way in which this cooperation is accomplished is by a process known as quorum sensing or autoinduction. This is a phenomenon in which bacteria monitor their own population density through sensing the levels of signal molecules, sometimes called autoinducers because they can stimulate the cell that releases them. The concentration of these signal molecules increases along with the bacterial population until it rises to a specific threshold and signals the bacteria that the population density has reached a critical level or quorum. The bacteria then begin expressing sets of quorum-dependent genes. Quorum sensing has been found among both gram-negative and gram-positive bacteria. Quorum sensing makes good practical sense. Take the production and release of extracellular enzymes as an example. If such enzymes were released by only a few bacteria, they would diffuse away and be rendered ineffective because of dilution. With by quorum sensing, the bacteria reach a high population density before they release enzymes, and as a consequence enzyme levels are concentrated enough to have significant effects. This is an advantage within a host's body as well as in the soil or an aquatic habitat. If a pathogen levels at a particular site before producing virulence factors and escaping into surrounding host tissues, it has a much better chance of counteracting host defenses and successfully spreading throughout the host's body. This explains another pattern in quorum sensing. It seems to be very important in many bacteria that establish symbiotic or parasitic relationships with hosts. Quorum sensing was first discovered in gram-negative bacteria and is best understood in these microorganisms. The most common signals in gram-negative bacteria are acylhomoserine lactones (HSLs). These are small molecules composed of a 4 to 14 carbon acyl chain attached by an amide bond to homoserine lactone, figure 8a. The acyl chain may have a keto group or hydroxyl group on its third carbon. Acyl HSLs diffuse into the target cell, figure 8b. Once they reach a sufficiently high level, acyl HSLs bind to special receptor proteins and trigger a conformational change. Usually the activated complexes act as inducers, that is, they bind to target sites on the DNA and stimulate transcription of quorum-sensitive genes. The gene needed to synthesize acyl HSL is also produced frequently, thus amplifying the effect by the production and release of more autoinducer molecules. Many different processes are sensitive to acyl HSL signals in quorum sensing in gram-negative bacteria. Some well-studied examples: bioluminescence production by Vibrio fischeri, 2 Pseudomonas aeruginosa synthesis and release of virulence factors, 3 conjugal transfer of genetic material by Agrobacterium tumefaciens, and 4 antibiotic production by Erwinia carotivora and Pseudomonas oreofaciens. Gram-positive bacteria also regulate activities by quorum sensing, often using an oligopeptide signal. Good examples are mating in Enterococcus faecalis, competence induction in Streptococcus pneumoniae, stimulation of sporulation by Bacillus subtilis, and production of many toxins and other virulence factors by Staphylococcus aureus. Quorum sensing even stimulates the development of aerial mycelia and the production of streptomycin by Streptomyces griseus. In this case, the signal seems to be gamma-butyrolactone rather than an oligopeptide. An interesting and important function of quorum sensing is to promote the formation of mature biofilms by the pathogen Pseudomonas aeruginosa, and it may play a role in cystic fibrosis. Biofilm formation makes sense for the pathogen because biofilms protect against antibiotics and detergents. Quorum sensing should be very effective within biofilms because there will be less dilution and acyl HSL levels will increase rapidly. Under circumstances, two different bacteria might stimulate each other by releasing similar signals. This appears to be the case in biofilms containing the pathogens P. aeruginosa and Burkholderia capacia. Quorum sensing is an example of what might be called multicellular behavior in that many individual cells communicate and coordinate their activities to act as a unit. Other examples of such complex behavior are pattern formation in colonies and fruiting body formation in the myxobacteria. Unit -3. Fundamental processes. Institute for Advanced Studies, Jodhpur 1. Institute for Advanced Studies, Jodhpur 1. A1. DNA replication, unit of replication, enzymes involved, replication origin and replication fork, fidelity of replication. DNA replication in prokaryotes. DNA replication is carried out by enzymes called DNA polymerases using deoxyribonucleoside triphosphates, DNTPs, as precursors. The process starts with the separation of the two strands of the DNA helix to expose the bases. New strands of DNA are synthesized using the old strands as templates. Complementary base pairing between the bases on the incoming nucleotides and the template DNA strand A, T, G, C dictates which nucleotide will be inserted into the growing DNA chain. Semi-conservative replication is, after the whole DNA molecule has been copied, each new DNA helix consists of one original DNA strand and one newly synthesized DNA strand. The role of the DNA polymerase is to check that the hydrogen bonding is correct and then to form a phosphodiester bond between two adjacent nucleotides. As the basic mechanism for DNA replication is the same for all organisms, this will be described first in relation to bacterial DNA replication. Differences found in eukaryotes will be discussed subsequently. Origin of replication. The first stage of replication involves the identification of the origin of replication. There is only one on the chromosome of bacteria called ORAC. A complex of proteins binds to this site, opens up the helix and initiates replication. DNA synthesis occurs in both directions, bidirectional, and on both strands, creating a replication bubble which appears as a theta-theta structure in electron micrographs. The two sites at which DNA synthesis occurs are called the replication forks. As replication proceeds, the replication forks move around the molecule, opening up the DNA strands, synthesizing two new complementary strands and rewinding up the DNA behind until the two replication forks meet at a termination site. The two completed circles of DNA are still linked and have to be separated by an enzyme topoisomerase II, which makes a transient break in both strands of one of the molecules, allowing them to disconnect. Other enzymes, proteins associated with DNA replication are DNA helicase, which unwinds the DNA helix to allow access to the bases. Single-stranded binding SSB proteins that bind to the DNA strands, preventing them from base pairing before they have been copied. There is a problem in that as the helix is opened up, the DNA ahead of the replication fork would become overwound because the DNA ends are fixed. The chromosome is circular and therefore cannot rotate relative to each other. This is solved by topoisomerase I, which makes a break in one of the DNA strands, which allows the DNA to rotate round the intact strand, thus releasing the torsion. DNA polymerase, enzyme responsible for normal DNA synthesis in bacteria, is DNA polymerase III. DNA polymerase I also has a role in DNA replication and is the enzyme that is involved in DNA repair. The role of DNA polymerase II is unknown as yet. Enzymes function by adding a new nucleotide to the 3' end of a growing polynucleotide chain with the release of pyrophosphate, synthesizing a new strand of DNA in a 5' 3' direction. Institute for Advanced Studies, Jodhpur 2. DNA fidelity proofreading. DNA polymerases also have exonuclease activity, which allows the sequential removal of nucleotides in both the 5' 3' and 3' 5' directions. The ability to remove indirectly inserted nucleotides confers proofreading properties on DNA polymerase III. After the enzyme has inserted a nucleotide into the ring chain, the enzyme can detect if the base pairing is correct or if a mismatch has occurred. There is a mismatch. The 3' 5' exonuclease activity removes the nucleotide and the polymerase can then replace it with the correct one. This proofreading ability ensures that the error rate, mutation rate, in synthesis is therefore less than the 10th to the 8th of October 10 per base pair. There are several features of DNA replication that are due to the nature of all polymerase enzymes. 1. DNA polymerases cannot start from scratch. They must have a 3' group to add a nucleotide onto. These enzymes therefore require some sort of primer to start DNA synthesis. 2. DNA can only be synthesized in one direction, 5' 3'. As the two strands DNA are antiparallel and are both copied at the same time at the replication fork, only one strand can be synthesized continually. The other strand must be synthesized in short fragments. Primes. The problem of how to start DNA synthesis is solved by using a specialist RNA polymerase called a primase to lay down a short stretch of ribonucleotides primer complementary to the DNA template at the start point for DNA synthesis. RNA polymerases do not require a 3' group to start RNA synthesis, so can start from scratch. Short piece of RNA provides the necessary 3' group to which DNA polymerase L can join a deoxyribonucleotide and start to synthesize the DNA strand. Synthesis continues until the DNA polymerase III reaches a primer. DNA polymerase III is released and DNA polymerase I binds in its place. DNA polymerase I has a 3' 5' exonuclease activity, which confers the ability to remove the ribonucleotides one by one, while its polymerase activity allows it to replace these with deoxyribonucleotides. Gap in the deoxyribonucleotide chain is sealed by a specialist enzyme, DNA ligase, which can join adjacent 5' phosphate and 3' groups. As DNA can only be synthesized in the 5' 3' direction, only one strand can be synthesized continuously. This is called the leading strand. The other strand, called the lagging strand, is synthesized as short pieces, approximately 1,000 bases long, called Okazaki fragments. Therefore, constant reinitiation of DNA synthesis by a complex of proteins called the chromosome, as described in the section above, with the consequent joining of the fragments by the removal of the RNA primers by DNA polymerase I and sealing of the gaps by DNA ligase. Rolling circle replication. Institute for Advanced Studies, Jodhpur 3. For DNA replication is used by some bacteriophage and viruses and in the transfer of plasmid DNA between cells by conjugation called rolling circle replication. One strand of the DNA is nicked to provide 3' group. Helicase and SSB proteins create a replication fork and nicked strand is displaced. DNTPs are added to the 3' group to create the leading strand, and the displaced strand acts as a template for lagging strand synthesis. Eukaryotic replication. The differences between prokaryotic and eukaryotic replication are mainly as a result of the larger size of the genomes and the fact that they are linear. Some of the main ones are listed below. There are many origins of replication on one chromosome, approximately 1 every 3 to 300 kb of DNA. Leading and lagging strands are made by different DNA polymerases. The Okazaki fragments are shorter. Because of the need to reinitiate DNA synthesis constantly on the lagging strands, there are short segments at the ends, telomeres, which do not get copied. The chromosomes would therefore gradually shorten with time. This problem is remedied by a specialist RNA-containing enzyme, a telomerase, which extends the ends of chromosomes by adding numerous hexanucleotide repeat sequences. These then act as templates for DNA replication. Because of the complexities of chromatin structure, eukaryotic replication forks move at around 50 bp per sec. At the rate, it would take a few days to copy a typical mammalian chromosomal DNA molecule of 108 bp using 2 replication forks, hence the need for multiple replicons, typically 5,000, 1,000 in a mammalian cell. To study all these simultaneously would be a daunting prospect. Fortunately, the yeast Saccharomyces cerevisiae has a much smaller genome, 1.4 x 107 bp and 16 chromosomes, and only 400 replicons. The complete genome sequence is known. Simpler still are viruses such as simian virus 40, SV40. SV40 DNA is a 5 kilobits double-stranded circle which forms nucleosomes when it enters the cell nucleus. It provides an excellent model for a mammalian replication fork. Another useful system is a cell-free extract prepared from the eggs of the African clod frog, Xenopus laevis. Because of its high concentration of replication proteins sufficient for several cell doublings in vivo, this extract can support the extensive replication of added DNA or even whole nuclei. Origin and initiation. In eukaryotes, clusters tandem arrays of about 20 to 50 replicons initiate simultaneously at defined times throughout S phase. Those replicate early in S phase comprise predominantly euchromatin, which includes transcriptionally active DNA, while those activated late in S phase are mainly within heterochromatin. Centromeric and telomeric DNA replicates last. This pattern reflects the accessibility of the different chromatin structures to initiation factors. Individual yeast replication origins have been cloned into prokaryotic plasmids. Since they allow these plasmids to replicate in yeast, a eukaryote, they are termed autonomously replicating sequences, ARSs. The minute that will support replication is only 11 bp and has a consensus sequence ATTTT8GTTT8T, though additional copies of the sequence are required for optimal efficiency. This sequence is bound by the origin recognition complex, ORC, which, when activated by CDcase, permits opening of the DNA for copying. Giant origin sequences have not been isolated from mammalian cells, and it is believed that initiation of each replicon may occur at random within an initiation zone which may be several kilobases in length and which may be part of the dispersed repetitive DNA fraction. In contrast to prokaryotes, eukaryotic replicons can only initiate once per cell cycle. A proteasing factor, which is absolutely required for initiation and inactivated after use, can only gain access to the nucleus when the nuclear envelope dissolves at mitosis, thus preventing premature reinitiation. Replication fork. Before copying, the DNA must be unwound from the nucleosomes at the replication forks. This slows the movement of the forks to about 50 bp per sec. After the fork has passed, new nucleosomes are assembled from Institute for Advanced Studies, JADPR 4. A mixture of old and newly synthesized histones. As in karyotes, one or more DNA helicases in a single-stranded binding protein, replication protein A, RPA, are required to separate the strands. 3 different DNA polymerases are involved in elongation. The leading strands in each lagging strand fragment are initiated with RNA by a primase activity that is an integral part of DNA polymerase alpha. This polymerase continues elongation with DNA but is quickly replaced by DNA polymerase delta on the leading strand and probably also on the lagging strand. The role of DNA polymerase epsilon is less clear, but it may simply complete the lagging strand fragments after primer removal. Both these enzymes have associated proofreading activity. Synthesized long DNA is conferred on DNA polymerase delta by proliferating cell nuclear antigen, pCNA, the functional equivalent of the beta subunit of E. coli DNA polymerase III holoenzyme. Small size of eukaryotic lagging strand fragments, e.g. 135 bp and SV40, appears to reflect the amount of DNA unwound from each nucleosome as the fork progresses. In addition to doubling the DNA, the histone content of the cell is also doubled during S phase. Nuclear matrix. The nuclear matrix is a scaffold of insoluble protein fibers which acts as an organizational framework for nuclear processes, including DNA replication. Huge replication factories containing all the enzymes and DNA associated with the replication forks of all replicons within a cluster are immobilized on the matrix, until the DNA moves through these sites as it replicates. These factories can be visualized in the microscope by pulse labeling the replicating DNA with the thymidine analog, bromodeoxyuridine BUDR, and visualizing the labeled DNA by immunofluorescence using an antibody that recognizes BUDR. Telomeric replication. Ends of linear chromosomes cannot be fully replicated by semi-discontinuous replication as there is no DNA to elongate to replace the RNA removed from the 5' end of the lagging strand. Thus, genetic information could be lost from the DNA. To overcome this, the ends of eukaryotic chromosomes consist of hundreds of copies of a simple, non-informational repeat sequence e.g. TTAGGG in humans with the 3' end overhanging the 5' end. 3' AATCCCAATCCC5. 5' TTAGGGT. The enzyme telomerase contains a short RNA molecule, part of whose sequence is complementary to this repeat. This RNA acts as a template for the addition of these repeats to the 3' overhang by repeated cycles of elongation, polymerization, and translocation. The complementary strand is then synthesized by normal lagging strand synthesis, leaving a 3' overhang. Interestingly, telomerase activity is repressed in the somatic cells of multicellular organisms, resulting in a gradual shortening of the chromosomes with each cell generation. As this shortening reaches informational DNA, the cell senesces and die. This phenomenon may be important in cell aging. Furthermore, the unlimited proliferative capacity of many cancer cells is associated with reactivation of telomerase activity. Institute for Advanced Studies, JADPR 5. 2. Extrachromosomal replicons. Plasmid replication and maintenance. Copy number. Plasmid replication and maintenance is a totally intrinsic property applicable to all plasmids, allowing them to be classified according to replication and partition strategy and how their copy number is regulated. Plasmid replication is autonomous, but may be coupled to the replication of the host genome and may be influenced by the host and its environment. Generally, plasmids require use of the host-encoded replication machinery, e.g. DNA polymerase, RNA polymerase, DNA primase, but encode other factors required for the initiation of replication and its regulation. Initiation occurs at a specific origin and is the rate-limiting step in plasmid replication. The frequency of successful initiation establishes the characteristic average number of plasmid molecules per cell, the copy number. In bacteria, larger plasmids usually have a copy number of 1 to 3. Their replication is coupled to that of the chromosome. These are termed single-copy plasmids and they are usually conjugated. Smaller plasmids have higher copy numbers, typically 10 to 30, and are termed multi-copy plasmids. Institute for Advanced Studies, JADPR 6. Replication occurs randomly and is self-regulated. The plasmids are usually non-conjugative. The replication control mechanisms which maintain the correct copy number do so by measuring the relative concentration of origins in the cell. If a mutation disrupts the function of a key regulator, the copy number can change substantially. Severely repressed replication can lead to plasmid loss, and in regulated replication, runaway replication, can increase the copy number tenfold. Runaway replication is capped only when another component, e.g. an enzyme or substrate for DNA replication, becomes limiting. A plasmid carrying such a mutation is known as a COP mutant. The behavior of eukaryotic plasmids has been studied in the Yeast S. cerevisiae. Plasmids with the 2' origin are maintained at high copy number, less than 100, although replication occurs only in the S phase and is coupled to genome replication. Conversely, plasmids which resemble chromosomes, i.e. those possessing Rs origins in a centromere, are maintained at a copy number of 1 to 2. Centromeres dominant to plasmid origins, so that artificial plasmids containing centromeres, ARSs and 2' origins behave like chromosomes, yeast cloning vectors. Plasmid maintenance. As well as controlling self-transfer and replication, plasmids also encode maintenance functions, ensuring both daughter cells inherit plasmids following cell division. The distribution of plasmids to the daughters of a dividing cell is partition. Without maintenance, spontaneous curing, table 1, can occur during partition. Cells without plasmids are more committed than plasmid-containing cells under normal growth conditions, i.e. in the absence of selection for the phenotype conferred by the plasmid, because they do not divert their resources to plasmid functions. Therefore, cured cells rapidly increase in the population at the expense of the plasmid-containing cells. Maintenance functions have been well characterized in bacteria and fall into several groups, table 2. The existence of maintenance functions places plasmids, along with viruses and transposable elements, within the category of selfish DNA. Plasmid compatibility. Incompatibility is the inability of two plasmids to coexist in the same host strain unless conditions are imposed which select for the phenotype conferred by both plasmids. Incompatible plasmids rapidly segregate in a growing unselected population to yield two homoplasmid strains. Incompatibility occurs between plasmids with similar strategies for the control of replication and/or partition. The control plasmid copy number is predominantly by negative regulation, thus two distinct single copy number is predominantly by negative regulation, thus two distinct single copy plasmids with the same regulatory mechanism in the same cell will repress each other's replication until cell division, when they become segregated and repression is lifted. Mixed multicopy plasmids also demonstrate mutual repression until cell division. In the daughter cells, repression is lifted, and because multicopy plasmid replication is random, each type of plasmid has an equal chance to undergo replication. The two seeds will then achieve a higher copy number and will be at an advantage at the next round of division, eventually leading to the generation of homoplasmid cells. Multicopy plasmids thus take longer to Institute for Advanced Studies, JODPR 7. Segregate than single copy plasmids. Single copy plasmids with the same partition mechanism segregate due to competition for the same partition site, which is presumably represented only twice in a cell. Plasmid incompatibility provides a useful system of classification according to replication and partition strategy. Mutant incompatible plasmids, i.e. those with similar strategies, are placed into an incompatibility group, in group of which there are approximately 30 for E. coli and related enterobacteria. Mechanisms of plasmid DNA replication. Closed circular double-stranded DNA plasmids of bacteria replicate similarly to the chromosome, with initiation characterized by the binding of specific initiation proteins called rep proteins to repetitive elements at the plasmid origin. For many plasmids, e.g. R1 rep facilitates unwinding of the origin, allowing the loading of helicase and the establishment of either a single replication fork or two replication forks. In other plasmids, e.g. PT181 of Staphylococcus, the rep protein is a nickase which initiates rolling circle replication. Col-1-related plasmids do not require rep proteins because the host RNA polymerase is used to transcribe through the origin to generate a primer for the leading strand. Such plasmids are thus under relaxed control. Linear bacterial plasmids are similar to linear virus genomes, e.g. they possess covalently sealed ends, e.g. Borrelia plasmids, or terminal proteins and inverted repeats, e.g. plasmids, and may replicate using the same strategies employed by viruses. Linear plasmids of eukaryotic organelles often encode their own polymerases, which are presumably utilized for autonomous replication. The discussion below relates to strategies for the regulation of replication unique to plasmids. Multiple origins and iterns. Many larger plasmids have multiple origins, often associated with different control mechanisms, making it difficult to assign such plasmids to single incompatibility groups. In plasmids, e.g. the F plasmid, one particular origin is favored, and the role of the extra origins is unknown. In others, e.g. R6K, the alternative origins are used with equal frequency. Many recombinant plasmid vectors have been deliberately designed to incorporate both prokaryotic and eukaryotic origins, allowing them to be maintained in both types of cells. Shuttle vector. Plasmid origins are often characterized by essential repetitive sequences termed iterns. These are common motifs in plasmids, e.g. plasmid RK2, and in some phages which can replicate like plasmids, e.g. bacteriophages P1 and lambda, as well as the bacterial chromosome itself. Origin of replication. Plasmid iterns are binding sites for the rep proteins, which may act alone or may associate with host initiator proteins, e.g. DNA and E. coli. In plasmid RK6, for instance, regardless of which of the three origins is used, the sequence of 7 direct tandem repeats located within the gamma origin is essential for replication. Control of plasmid replication by antisense RNA. Plasmid L-type origin of replication, including the vast majority of plasmid cloning vectors, initiate leading strand DNA synthesis from a single RNA primer generated by the host RNA polymerase primer, priming strategy. The origin is actually transcribed on both strands to yield two transcripts, RNAi and RNA2. RNA2 is the primer, whereas RNAi, which is complementary to part of RNA2, acts as a repressor by sequestering RNA2 into an inactive duplex. DNA synthesis begins the origin, but transcription of RNA2 begins 555 BP upstream of the origin and continues through it and beyond. The transcript must therefore be processed by cleavage at the origin to yield a functional primer. This processing is carried out by RNA-SEH nucleases and is facilitated by the three-stem loop secondary structure adopted by RNA2. Interaction between RNAi and RNAi1 culminates in their full hybridization, which disrupts the secondary structure of RNA2 and prevents it hybridizing to the DNA. This in turn prevents the cleavage reaction and results in a replication block. In vitro, RNAi is able to anneal to RNA2 only when the latter is between 100 and 360 nt in length. This means that RNAi must hybridize to the nascent RNA2 transcript during the early phase of its synthesis to repress replication. Plasmid-encoded protein called ROM enhances the rate of RNAi, RNA2 hybrid formation once RNA2 is greater than 200 nucleotides in length and is thus a key regulator of Col1 replication. The replication of the R1 plasmid is also regulated by antisense RNA. The messenger protein, REPA, is essential for the initiation of replication, but transcription of an adjacent gene generates an antisense RNA COPA, which binds to the leader region of the REPA mRNA designated COPT to prevent its translation. Antisense RNA, translational control. The COPA and COPT RNAs both form secondary structures, and mutational analysis has shown that the interaction between them may involve these secondary structures rather than full duplex formation, a so-called kissing complex. The prevalence of antisense RNA regulators in plasmids suggests that RNA offers a unique strategy for replication control for which protein is insufficient. In many diverse plasmids the same mechanism occurs. A short antisense inhibitory factor anneals to the 5' end of an essential functional RNA. Institute for Advanced Studies, JODPR 8. And blocks its activity. Possible explanation is that the maintenance of plasmid copy number requires an unstable regulator so that the plasmid population can respond rapidly to deviations from a standard copy number. RNA is a suitable candidate for such an unstable regulator, antisense RNA. Relaxed and stringent control. Plasmid replication may be under relaxed or stringent control. If cellular protein synthesis is inhibited, for example by chloramphenicol, some plasmids continue to replicate and their copy number increases above normal vegetative levels. These are relaxed plasmids, and are typically small multicopy plasmids. Other plasmids cease to replicate along with the host chromosome when de novo protein synthesis is inhibited. These are described as stringent plasmids, and are typically large, single-copy, conjugative plasmids. Most general-purpose plasmid cloning vectors are under relaxed control. The ability of relaxed plasmids to continue to replicate in the absence of host protein synthesis reflects the stability of host proteins required for the initiation of replication, and the instability of plasmid-encoded regulators. For instance, plasmid col-1 is under relaxed control because the initiation factor ROM has a negative role. It prevents initiation by facilitating the pairing of an antisense RNA with the leading strand primer. In the absence of ROM, plasmid replication is repressed and copy number increases. Very high number cloning vectors have no functional ROM gene. Conversely, stringent plasmids such as F require a positively acting rep protein for initiation. In the absence of rep, plasmid replication is blocked regardless of the availability of host DNA replication proteins. A3. DNA damage and their role in carcinogenesis. The DNA sequence can be changed as the result of copying errors introduced by DNA polymerases during replication and by environmental agents such as mutagenic chemicals and certain types of radiation. If the sequence changes, whatever their cause, or left undirected, both growing and non-growing somatic cells might accumulate so many mutations that they could no longer function. In addition, the DNA in germ cells might incur too many mutations for viable offspring to be formed. Thus, the correction of DNA sequence errors in all types of cells is important for survival. The presence of DNA damage and repair to the generation of cancer carcinogenesis became evident when it was recognized that all agents that cause cancer carcinogens also cause a change in the DNA sequence and thus are mutagens. Effects of carcinogenic chemicals on tumor production can be accounted for by the DNA damage that they cause and by the errors introduced into DNA during the cell's efforts to repair this damage. UV, radiation and ionizing radiation, X-rays and atomic particles not only modify DNA, but also can cause cancer in animals and can transform normal cells in culture into rapidly proliferating, cancer-type cells. The ability of radiation to cause human cancer, especially leukemia, was dramatically shown by the increased rates of leukemia among survivors of the atomic bombs dropped in World War II, and more recently by the increase in melanoma, skin cancer, in individuals exposed to too much sunlight. Lesion. A lesion is an alteration to the normal chemical or physical structure of the DNA. Some of the hydrogen and carbon atoms in the heterocyclic ring systems of the bases and some of the exocyclic functional groups, i.e. the keto and amino groups of the bases, are chemically quite reactive. Many exogenous agents, such as chemicals and radiation, can cause changes to these positions. The altered chemistry of the bases may lead to loss of base pairing or altered base pairing, e.g. an altered A may base pair with C instead of T. If the lesion was allowed to remain in the DNA, a mutation could become fixed in the DNA by direct or indirect mutagenesis. Alternatively, the chemical change may produce a physical distortion in the DNA which blocks replication and/or transcription, causing cell death. Thus, DNA lesions may be mutagenic and/or lethal. Some spontaneous and occur because of the inherent chemical reactivity of the Some lesions are spontaneous and occur because of the inherent chemical reactivity of the DNA in the presence of normal, reactive chemical species within the cell. For example, the base cytosine undergoes spontaneous hydrolytic deamination to give uracil Pig. If left unrepaired, the resulting uracil would form a base pair with adenine during subsequent replication, giving rise to a point mutation. In fact, the generation of uracil in DNA in this way is the probable reason why DNA contains thymine instead of uracil. Any uracil found in DNA is removed by an enzyme called uracil DNA glycosylase and is replaced by cytosine. 5-methylcytosine, a modified base found in small amounts in DNA, deaminates to thymine, a normal base. This is much more difficult to detect. Deamination is another spontaneous hydrolytic reaction that involves cleavage of the N-glycosylic bond between N9 of the purine bases A and G and Cl, of the deoxyribose sugar and hence loss of purine bases from the DNA. The sugar phosphate backbone of the DNA. Institute for Advanced Studies, Jodhpur 9. Remains intact. The resulting apurinic site is a non-coding lesion, as information encoded in the purine bases is lost. Depurination occurs at the rate of 10,000 purines lost per human cell per hour at 37 degrees Celsius. Though less frequent, depurimidination can also occur. Oxidative damage. This occurs under normal conditions due to the presence of reactive oxygen species, Ru, in all aerobic cells, for example superoxide, hydrogen peroxide and Oxidative damage. This occurs under normal conditions due to the presence of reactive oxygen species, Ru, in all aerobic cells, for example superoxide, hydrogen peroxide and, most importantly, the hydroxyl radical O. This radical can attack DNA at a number of points, producing a range of oxidation products with altered properties, for example 8-oxoguanine, 2-oxodenine and 5-formyl Pig. The levels of these can be increased by hydroxyl radicals from the radiolysis of water caused by ionizing radiation. Alkylating agents are electrophilic chemicals which readily add alkyl, e.g. methyl, groups to various positions on nucleic acids distinct from those methylated by normal methylating enzymes. Common examples are methylmethane sulfonate, MMS, and ethyl nitrosourea, ENU, Pig. Typical examples of methylated bases are 7-methylguanine, 3-methyladenine, 3-methylguanine and 0,6-methylguanine, Pig. Some of these lesions are potentially lethal as they can interfere with the unwinding of DNA during replication and transcription. Also indirectly mutagenic. However, 0,6-methylguanine is a directly mutagenic lesion as it can base pair with thymine during replication. Cyclobutane pyrimidine dimers are formed by ultraviolet light from adjacent pyrimidines on one strand by CYC1ization of the double-bonded C5 and C6 carbon atoms of each base to give a cyclobutane ring, Pig. The result of base pairing with the opposite strand causes localized denaturation of the DNA, producing a bulky lesion which would disrupt replication and transcription. Another pyrimidine dimer, the 6,4 photoproduct, results from the formation of a bond between C6 of one pyrimidine base and C4 of the adjacent base, C-ring carbon numbers in Pig. Detoxification begins with a powerful series of oxidation reactions catalyzed by a set of proteins called cytochrome P450. These enzymes, which are bound to endoplasmic reticulum membranes, can oxidize even highly unreactive compounds such as polycyclic aromatic hydrocarbons. Oxidation of polycyclic aromatics produces an epoxide, a very reactive electrophilic group. These epoxides are rapidly hydrolyzed into hydroxyl groups, which are then coupled to glucuronic acid or other groups, producing compounds soluble enough in water to be excreted. Some intermediate epoxides, however, are only slowly hydrolyzed to hydroxyl groups, probably because the relevant enzyme epoxide hydratase cannot get to the epoxide to act on it. For example, the indirect acting carcinogen benzo of pyrene undergoes 2 oxidation reactions to yield a highly reactive electrophilic ultimate carcinogen. Other types of indirect acting carcinogens are activated by different oxidative pathways, which also involve P450 enzymes. The liver carcinogen aflatoxin B1 also covalently binds to DNA. Institute for Advanced Studies, Jodhpur 10. A4. DNA repair mechanisms. During DNA replication and DNA damage due to exposure to mutagens happen all the time, and microorganisms have evolved very complex systems to recognize and repair the damage. The repair directly undoes the original damage, causing no change to the base sequence, but sometimes the amount of damage is so great that DNA synthesis cannot continue. In addition to the proofreading activity of DNA polymerases that can correct miscopied bases during replication, cells have evolved mechanisms for repairing DNA damage by chemicals or radiation. Complex organisms with large genomes and relatively long generation times contain many cells that divide very slowly or not at all, e.g., liver and brain cells. Such cells must use the information in their DNA for weeks, months, or even years, greatly increasing their chances for sustaining damage to their DNA. If repair processes were 100% effective, chemicals and radiation would pose no threat to cellular DNA. Fortunately, repair of lesions caused by some environmental agents is relatively inefficient, and such lesions can lead to mutations that ultimately cause cancer. In theory, a carcinogen could act by binding to DNA and causing a change in the sequence that is perpetuated during DNA replication. Current evidence suggests, however, that many permanent DNA sequence changes are induced by the very repair processes cells use to rid themselves of DNA damage. 4.1. Direct reversal. A. Photoreactivation. Cyclobutane pyrimidine dimers can be monomerized again by DNA photolysis, photoreactivating enzymes, in the presence of visible light. These enzymes have prosthetic groups which absorb blue light and transfer the energy to the cyclobutane ring, which is then cleaved. The E. coli photolyase has two chromophores, N5, N10-methynyl tetrahydrofolate and reduced flavin-adenine dinucleotide, FADH. Photoreactivation is specific for pyrimidine dimers. It is an example of direct reversal of a lesion and is error-free. B. Alkyltransferase. Another example of error-free direct reversal forms part of the adaptive response to alkylating agents. This response is induced in E. coli by low levels of alkylating agents and gives increased protection against the lethal and mutagenic effects of subsequent high doses. Mutagenic protection is afforded by an alkyltransferase which removes the alkyl group from a directly mutagenic O6-alkylguanine. Curiously, the alkyl group is transferred to the protein itself and inactivates it. Thus, each alkyltransferase can only be used once. This protein is also present in mammalian cells. Protection against lethality involves induction of a DNA glycosylase which removes other alkylated bases through base excision repair. 4.2. Excision repair mechanism. This ubiquitous mechanism operates on a wide variety of lesions and is essentially error-free. There are two forms, nucleotide excision repair (NER) and base excision repair (BER). NER, an endonuclease cleaves the DNA a precise number of bases on either side of the lesion and an oligonucleotide containing the lesion is removed, leaving a gap. For example, in E. coli, the UVR-ABC endonuclease removes pyrimidine dimers and other bulky lesions by recognizing the distortion these produce in the double helix. In BER, the 5 bases are recognized by relatively specific DNA glycosylases which cleave the N-glycosylic bond between the altered base and the sugar Cu, leaving an apurinic or a pyrimidinic AP site. AP sites are also produced by spontaneous base loss. An AP endonuclease then cleaves the DNA at this site and a gap may be created by further exonuclease activity. The gap is generally larger in NER and can be as small as 1 nucleotide in BER. From this point on, both forms of excision repair are essentially the same. In coli, the gap is filled by DNA polymerase I and the final phosphodiester bond made by DNA ligase, much as in the final stages of processing of the lagging strand fragments during DNA replication. In eukaryotes, gap filling in BER involves predominantly DNA polymerase beta, whereas the Institute for Advanced Studies, JADBR 11. Longer gaps generated in NER are filled by DNA polymerases delta or epsilon. In eukaryotic NER, recognition and excision of DNA damage is a complex process, involving at least 18 polypeptide factors, including the transcription factor TFIIH. Excision is coupled to transcription so that transcribed, genetically active regions of the DNA are repaired more rapidly than non-transcribed DNA. This helps to limit the production of defective gene products. 4.3. Mismatch repair. These spontaneous mutations are point mutations, which involve a change in a single base pair in the DNA sequence. These can arise from errors in replication, during genetic recombination, and, particularly, by base deamination whereby AC residue is converted into AU residue. Real and eukaryotic cells have a mismatch repair system that recognizes and repairs all single base mispairs except C bullet operator C, as well as small insertions and deletions. The conceptual problem with mismatch repair is determining which is the normal and which is the mutant DNA strand, and repairing the latter so that it is properly base paired with the normal strand. In DNA, adenine residues in a GATC sequence are methylated at the 6th position. Since DNA polymerases incorporate adenine, not methyladenine, into DNA, adenine residues in newly replicated DNA are methylated only on the parental strand. The adenines in Institute for Advanced Studies, JADBR 12. GTC sequences on the daughter strands are methylated by a specific enzyme, called DAM-methyltransferase, only after a lag of several minutes. During this lag period, the newly replicated DNA contains hemimethylated GATC sequences. An E. coli protein designated MUth, which binds specifically to hemimethylated sequences, is able to distinguish the methylated parental strand from the unmethylated daughter strand. If an error occurs during DNA replication, resulting in a mismatched base pair near a GATC sequence, another protein, MUTS, binds to this abnormally paired segment. Binding of MUTS triggers binding of MUT-L, a linking protein that connects MUTS with a nearby MUth. This linking activates the latent endonuclease activity of MUth, which then cleaves specifically the unmethylated daughter strand. Following this initial incision, the segment of the daughter strand containing the MIS incorporated base is excised and replaced with the correct DNA sequence. E. coli strains that lack the MUTS, MUth, or MUT-L protein have a higher rate of spontaneous mutations than wild-type cells. Strains that cannot synthesize the DAM-methyltransferase also have a high rate of spontaneous mutations. Because DAM strains cannot methylate adenines within GATC sequences, the MUT-HLS mismatch repair system cannot distinguish between the templated and newly synthesized strand and therefore cannot efficiently repair mismatched bases. A similar mechanism repairs lesions resulting from depurination, the loss of a guanine or adenine base from DNA resulting from cleavage of the glycosidic bond between deoxyribose and the base. Depurination occurs spontaneously and is fairly common in mammals. The resulting apurinic sites, if left unrepaired, generate mutations during DNA replication because they cannot specify the appropriate paired base. All cells possess apurinic AP endonucleases that cut a DNA strand near an apurinic site. As with mismatch repair, the cut is extended by exonucleases, and the resulting gap then is repaired by DNA polymerase and ligase. The discriminatory mechanism in eukaryotes is not known, but mismatch repair is clearly important. In maintaining overall error rate of DNA replication and, therefore, the spontaneous mutation rate, hereditary non-polyposis carcinoma of the colon is caused by mutational loss of one of the human mismatch repair enzymes. Mismatch repair may also correct errors that arise from sequence misalignments during meiotic recombination in eukaryotes. 4.4. Recombinational post-replication repair mechanism. Dimers cannot be replicated but, instead of stalling at these sites, DNA polymerase can jump over the lesion and resume DNA synthesis further down the DNA template. This leaves a gap in the newly synthesized DNA strand opposite the dimer. This cannot be repaired by excision repair as this requires an intact strand to be used as the template. Instead, the gap can be filled by REC-mediated recombination with the sister DNA helix, which contains the intact sequence that should be opposite the dimer pig. Though recombination does not repair the damage, it does create the situation where both new DNA molecules can be repaired by excision repair. Institute for Advanced Studies, Jodhpur 13. 4.5. Inducible DNA repair systems, SOS repair, are error-prone. Suffers so much DNA damage over a short time that its repair systems are saturated, it runs the danger of extensively replicating unrepaired lesions, thereby perpetuating mutations. In such situations, both bacterial and animal cells use inducible repair systems in an attempt to catch up. Such systems are not expressed in undamaged cells, but some aspect of the accumulated damage causes their derepression, induction, and expression. There are a number of genes and operons in the cell that are coordinately regulated by RECA protein and a repressor of transcription, LEXA. These are involved in dealing with DNA damage and are called SOS repair systems. These include an error-prone repair system which interacts with DNA polymerase, allowing it to continue replication of DNA past a pyrimidine dimer. The 3'5" proof reading ability of the enzyme is inhibited so that random bases can be inserted opposite the dimer without accurate base pairing. This mechanism is the main reason why UV light is mutagenic, because most of the other systems repair DNA accurately. The SOS repair systems are induced by RECA protein, activated by a conformation change, caused by the presence of damaged DNA. The activated RECA proteins causes LEXA protein to be proteolytically cleaved so that it no longer acts as a repressor of transcription. The genes in the system may then be expressed as long as the DNA damage is present in the cell. Once the damage is removed, RECA protein is no longer activated as an inducer of LEXA proteolysis, so LEXA protein accumulates in the cell and inhibits transcription of the SOS repair genes. Animal cells also have inducible repair systems, although it is not known whether these are error-prone. As noted earlier, however, the main mechanism for repairing double-strand breaks in eukaryotes clearly is error-prone. Double-strand repair and, perhaps, error-prone inducible repair likely play a role in mutagenesis and therefore in carcinogenesis in animals. In any case, many investigators believe that in animal cells, as in bacteria, most mutations are an indirect, not direct, consequence of DNA damage. Mutation or carcinogen-induced DNA damage must be repaired before the DNA is replicated. Cells have sensing mechanisms that react to DNA damage and stop DNA replication. These mechanisms, which are discussed in detail in the next chapter, involve checkpoint control proteins such as the p53 protein, which acts to stop the cell cycle if DNA is damaged, and thus to suppress production of tumors. Cells that do not express functional p53 protein exhibit high rates of mutation in response to DNA damage, accelerating the formation of tumors. Xeroderma pigmentosum (XP) is an autosomal recessive disorder characterized phenotypically by extreme sensitivity to sunlight and a high incidence of skin tumors. XP sufferers are defective in the NER of bulky DNA damage, including that caused by ultraviolet light. Defects in at least 7 different genes can cause XP, indicating the complexity of excision repair in mammalian cells. Xeroderma pigmentosum variant XPV is clinically very similar to classical XP, but cells from XPV individuals can carry out normal NER. In this case, the defect is in the gene encoding the translesion DNA polymerase eta, which normally inserts STAMP in an error-free fashion opposite the thymine residues in a cyclobutane-thymine dimer. XPV cells may, therefore, have to rely more heavily on alternative error-prone modes of translesion DNA synthesis to maintain DNA integrity after radiation damage. Sufferers of Cockayne syndrome are also sun-sensitive and defective in transcription-coupled excision repair, but are not cancer-prone. B1. Basic principles of transcription. Transcription is the enzymic synthesis of RNA on a DNA template. This is the first stage in the overall process of gene expression and ultimately leads to synthesis of the protein encoded by a gene. Transcription is catalyzed by an RNA polymerase which requires a dsDNA template as well as the precursor ribonucleotides ATP, GTP, ARN, UTP, Fig1. RNA synthesis always occurs in a fixed direction, from the 5' to the 3' end of the RNA molecule. Usually, only one of the 2 strands of DNA becomes transcribed into RNA. One strand is known as the sense strand. The sequence of the RNA is a direct copy of the sequence of the deoxynucleotides in the sense strand with U in place of T. The other strand is known as the antisense strand. This strand may also be called the template strand since it is used as a template to which ribonucleotides base pair for the synthesis of the RNA. Institute for Advanced Studies, Jodhpur 14. 1.1 Initiation of transcription. Initiation of transcription involves the binding of an RNA polymerase to the dsDNA. RNA polymerases are usually multi-subunit enzymes. They bind to the dsDNA and initiate transcription at sites called promoters Fig. 2. Promoters are sequences of DNA at the start of genes, that is to the 5' side upstream of the coding region. Sequence elements of promoters are often conserved between different genes. Differences between the promoters of different genes give rise to differing efficiencies of transcription initiation and are involved in their regulation. The short conserved sequences within promoters are the sites at which the polymerase or other DNA binding proteins bind to initiate or regulate transcription. In order to allow the template strand to be used for base pairing, the DNA helix must be locally unwound. Unwinding begins at the promoter site to which the RNA polymerase binds. The polymerase then initiates the synthesis of the RNA strand at a specific nucleotide called the start site, initiation site. This is defined as position +1 of the gene sequence Fig. 2. The RNA polymerase and its cofactors, when assembled on the DNA template, are often referred to as the transcription complex. 1.2 Elongation. The RNA polymerase covalently adds ribonucleotides to the 3' end of the growing RNA chain Fig. 1. The polymerase therefore extends the growing RNA chain in a 5' 3' direction. This occurs while the enzyme itself moves in a 3' 5' direction along the antisense DNA strand template. As the enzyme moves, it locally unwinds the DNA, separating the DNA strands, to expose the template strand for ribonucleotide base pairing and covalent addition to the 3' end of the growing RNA chain. The helix is reformed behind the polymerase. The E. coli RNA polymerase performs this reaction at a rate of around 40 bases per second at 37 degrees Celsius. 1.3 Termination. The termination of transcription, namely the dissociation of the transcription complex and the ending of RNA synthesis, occurs at a specific DNA sequence known as the terminator. Sequences often contain self-complementary regions which can form a stem loop or hairpin secondary structure in the RNA product Fig. 3. These cause the polymerase to pause and subsequently cease transcription. Other sequences can terminate transcription without the requirement for accessory factors, whereas other terminator sequences require the Rho protein Rho. Sequences can terminate transcription without the requirement for accessory factors, whereas other terminator sequences require the Rho protein Rho as an accessory factor. In the termination reaction, the RNA-DNA hybrid is separated allowing the reformation of the dsDNA, and the RNA polymerase and synthesized RNA are released from the DNA. B2. Transcription in prokaryotes E. coli 2.1 Escherichia coli RNA polymerase. The E. coli RNA polymerase is one of the largest enzymes in the cell. The enzyme consists of at least 5 subunits. These are the alpha, alpha, beta, beta, beta prime, beta, omega, W, and sigma, sigma subunits. In the complete polymerase called the holoenzyme, there are 2 subunits and 1 each of the other 4 subunits, i.e. alpha 2 BB, Os. The complete enzyme is required for transcription initiation. However, the sigma factor is not required for transcription elongation and is released from the transcription complex after transcription initiation. The remaining enzyme, which translocates along the DNA, is known as the core enzyme and has the structure alpha 2 BB, omega. E. coli RNA polymerase can synthesize RNA at a rate of around 40 nucleotide per second 37 degrees Celsius and requires Mg2+ for its activity. The enzyme has a non-spherical structure with a projection flanking a cylindrical channel. The size of the channel suggests that it can bind directly to 16 BP of DNA. The whole polymerase binds over a region of DNA covering around 60 BP. Although most RNA polymerases like the E. coli polymerase have a multi-subunit structure, it is important to note that this is not an absolute requirement. The RNA polymerase is encoded by bacteriophages T3 and T7 are single polypeptide chains which are much smaller than the bacterial multi-subunit enzymes. They synthesize RNA rapidly, 200 nt per c at 37 degrees Celsius, and recognize their own specific DNA binding sequences. Institute for Advanced Studies, Jodhpur 15. Alpha subunit. 2 identical alpha subunits are present in the core RNA polymerase enzyme. The subunit is encoded by the RPOA gene. The alpha subunit is required for core protein assembly, but has had no clear transcriptional role assigned to it. When phage T4 infects E. coli, the alpha subunit is modified by adenosine diphosphate (ADP) ribosylation of an arginine. This is associated with a reduced affinity for binding to promoters, suggesting that the alpha subunit may play a role in promoter recognition. Beta subunit. 1 beta subunit is present in the core enzyme. This subunit is thought to be the catalytic center of the RNA polymerase. Strong evidence for this has come from studies with antibiotics which inhibit transcription by RNA polymerase. The important antibiotic rifampicin is a potent inhibitor of RNA polymerase that blocks initiation but not elongation. This class of antibiotic does not inhibit eukaryotic polymerases and has, therefore, been used medically for treatment of gram-positive bacteria infections and tuberculosis. Rifampicin has been shown to bind to the beta subunit. Mutations that give rise to resistance to rifampicin map to RPOB, the gene that encodes the beta subunit. Further class of antibiotic, the streptolitogens, inhibit transcription elongation, and mutations that confer resistance to these antibiotics also map to RPOB. These studies suggest that the beta subunit may contain 2 domains responsible for transcription initiation and elongation. Beta subunit. 1 beta subunit is present in the core enzyme. It is encoded by the RPOC gene. This subunit binds to Mg2+ ions which are thought to participate in the catalytic function of the polymerase. A polyanion, heparin, has been shown to bind to the beta subunit. Heparin inhibits transcription in vitro and also competes with DNA for binding to the polymerase. This suggests that the beta subunit may be responsible for binding to the template DNA. Sigma factor. The most common sigma factor in E. coli is sigma 70, since it has a molecular mass of 70 kDa. Binding of the sigma factor converts the core RNA polymerase enzyme into the holoenzyme. The sigma factor has a critical role in promoter recognition, but is not required for transcription elongation. The sigma contributes to promoter recognition by decreasing the affinity of the core enzyme for nonspecific DNA sites by a factor of 104 and increasing affinity for the promoter. Many prokaryotes, including E. coli, have multiple sigma factors. They are involved in the recognition of specific classes of promoter sequences. The sigma factor is released from the RNA polymerase when the RNA chain reaches 8 to 9 nt in length. The core enzyme then moves along the DNA synthesizing the growing RNA strand. The sigma factor can then complex with a further core enzyme complex and reinitiate transcription. There is only 30% of the amount of sigma factor present in a cell compared with core enzyme complexes. Therefore, only 1/3 of the polymerase complexes can exist as holoenzyme at any one time. 2.2 The E. coli sigma 70 promoter. RNA polymerase binds to specific initiation sites upstream from transcribed sequence. These are called promoters. Although different promoters are recognized by different effectors which interact with the RNA polymerase core enzyme, the most common sigma factor in E. coli is sigma 70. Promoters were first characterized through mutations that enhance or diminish the rate of transcription of genes such as those in the lac operon. The promoter lies upstream of the start site of transcription, generally assigned as position +1. In accordance with this, promoter sequences are assigned a negative number reflecting the distance upstream from the start of transcription. Mutagenesis of E. coli promoters has shown that only very short conserved sequences are critical for promoter function. Promoter size. The sigma 70 promoter consists of a sequence of between 40 and 60 BP. region from around -55 to +20 has been shown to be bound by the polymerase, and the region from -20 to +20 is strongly protected from nuclease digestion by DNase. This suggests that this region is tightly associated with the polymerase, which blocks access of the nuclease to the DNA. Mutagenesis of promoter sequences show that sequences up to around position -40 are critical for promoter function. 2,6-BP sequences at around positions -10 and -35 have been shown to be particularly important for promoter function in E. coli. In sequence, the most conserved sequence in sigma-70 promoters is a 6-BP sequence which is found in the promoters of many different E. coli genes. This sequence is centered at around the -10 position with respect to the transcription start site Fig. 4. This is sometimes referred to as the Pribnow box, having been first recognized by Pribnow in 1975. It has a sequence of TATAAT, where the consensus sequence is made up of the most frequently occurring nucleotide at each position when many sequences are compared. The first two bases Ta and the final T are most highly conserved. This hexamer is separated by between 5 and 8 BP from the transcription start site. This intervening sequence is not conserved, although the distance is critical. The -10 sequence appears to be the sequence at which DNA unwinding is initiated by the polymerase. -35 sequence. Upstream regions around position -35 also have a conserved hexamer sequence, figure 4. This has a consensus sequence of TTGACA, which is most conserved in efficient promoters. The first three positions of this hexamer are the most conserved. This sequence is separated by 16 to 18 BP from the -10 box in 90% of all promoters. The intervening sequence between these conserved elements is not important. Institute for Advanced Studies, Jodhpur 16. Transcription start site. The transcription start site is a purine in 90% of all genes Fig. 4. G is more common at the transcription start site than often. There are C and T bases on either side of the start site nucleotide, i.e. CGT or CAT. Promoter efficiency. The sequences described above are consensus sequences typical of strong promoters. However, there is considerable variation in sequence between different promoters, and they may vary in transcriptional efficiency by up to 1,000-fold. Overall, the functions of different promoter regions can be defined as follows. The 35 sequence constitutes a recognition region which enhances recognition and interaction with the polymerase sigma factor. The -10 region is important for DNA unwinding. The sequence around the start site influences initiation. The sequence of the first 30 bases to be transcribed also influences transcription. Sequence controls the rate at which the RNA polymerase clears the promoter, allowing reinitiation of another polymerase complex, thus influencing the rate of transcription and hence the overall promoter strength. Importance of strand separation in the initiation reaction is shown by the effect of negative supercoiling of the DNA template, which generally enhances transcription initiation, presumably because the supercoiled structure requires less energy to unwind the DNA. Some promoter sequences are not sufficiently similar to the consensus sequence to be strongly transcribed under normal conditions. As is the lac promoter plaque, which requires an accessory activating factor called CAMP receptor protein (CRP) to bind to a site on the DNA close to the promoter sequence in order to enhance polymerase binding and transcription initiation. Most, such as those of genes associated with heat shock, contain different consensus promoter sequences that can only be recognized by an RNA polymerase which is bound to sigma factor different from the general factor sigma 70. 2.3. Initiation, elongation and termination of transcription. Promoter binding. The RNA polymerase core enzyme alpha-2 omega has a general nonspecific affinity for DNA. This is referred to as loose binding and it is fairly stable. When sigma factor is added to the core enzyme to form the holoenzyme, it markedly reduces the affinity for nonspecific sites on DNA by 20,000-fold. In addition, sigma factor enhances holoenzyme binding to correct promoter binding sites 100 times. Overall, this dramatically increases the specificity of the holoenzyme for correct promoter binding sites. The holoenzyme searches out and binds to promoters in the E. coli genome extremely rapidly. This is too fast to be achieved by repeated binding and dissociation from DNA, and is believed to occur by the polymerase sliding along the DNA until it reaches the promoter sequence. The polymerase recognizes the double-stranded -35 and -10 DNA sequences. The initial complex of the polymerase with the base paired promoter DNA is referred to as a closed complex. DNA unwinding. In order for the antisense strand to become accessible for base pairing, the DNA duplex must be unwound by the polymerase. Negative supercoiling enhances the transcription of many genes, since this facilitates unwinding by the polymerase. However, promoters are not activated by negative supercoiling, implying that differences in the natural DNA topology may affect transcription, perhaps due to differences in the steric relationship of the -35 and -10 sequences in the double helix. For example, the promoters for the enzyme subunits of DNA gyrase are inhibited by negative supercoiling. DNA gyrase is responsible for negative supercoiling of the E. coli genome, and so this may serve as an elegant feedback loop for DNA gyrase protein expression. The initial unwinding of the DNA results in formation of an open complex with the polymerase, and this process is referred to as tight binding. RNA chain initiation. Almost all RNA start sites consist of a purine residue, with G being more common, and unlike DNA synthesis, RNA synthesis can occur without a primer Fig. 5. The chain is started with a GTP or ATP, from which synthesis of the rest of the chain is initiated. The polymerase initially incorporates the first two nucleotides and forms a phosphodiester bond between them. The first 9 bases are added without enzyme movement along the DNA. After each one of these first 9 NT is added to the chain, there is a significant probability that the chain will be aborted. This process of abortive initiation is important for the overall rate of transcription since it has a major role in determining how long the polymerase takes to leave the promoter. Institute for Advanced Studies, Jodhpur 17. And allow another polymerase to initiate a further round of transcription. The minimum time for promoter clearance is 1 to 2 seconds, which is a long event relative to other stages of transcription. Chain elongation. When initiation succeeds, the enzyme releases the alpha factor and forms a ternary complex, 3 components, of polymerase to non-assent newly synthesized RNA, causing the polymerase to progress along the DNA promoter clearance, allowing reinitiation of transcription from the promoter by a further RNA polymerase holoenzyme. The region of unwound DNA, which is called the transcription bubble, appears to move along the DNA with the polymerase. The side region of unwound DNA remains constant at around 17 BP Fig. 6 and the 5' end of the RNA forms a hybrid helix of about 12 BP with the antisense DNA strand. This corresponds to just less than 1 turn of the RNA DNA helix. The E. coli polymerase moves at an average rate of 40 nt per sec, but the rate can vary depending on local DNA sequence. Maintenance of the short region of unwound DNA indicates that the polymerase unwinds DNA in front of the transcription bubble and rewinds DNA at its rear. The RNA-DNA helix must rotate each time a nucleotide is added to the RNA. RNA chain termination. The RNA polymerase remains bound to the DNA and continues transcription until it reaches a terminator sequence, stop signal, at the end of the transcription unit Fig. 7. The most common stop signal is an RNA hairpin in which the RNA transcript is self-complementary. As a result, the RNA can form a stable hairpin structure with a stem and a loop. Commonly the stem structure is very GC-rich, favoring its base pairing stability due to the additional stability of GC base pairs over AU base pairs. The RNA hairpin is often followed by a sequence of 4 or more U residues. It seems that the polymerase pauses immediately after it has synthesized the hairpin RNA. The subsequent stretch of U residues in the RNA base pairs only weakly with the corresponding residues in the antisense DNA strand. This favors dissociation of the RNA from the complex with the template strand of the DNA. The RNA is therefore released from the transcription complex. The base-paired antisense strand of the DNA then reanneals with the sense DNA strand and the core enzyme dissociates from the DNA. Rho-dependent termination. While the RNA polymerase can self-terminate at a hairpin structure followed by a stretch of U residues, other known terminator sites may not form strong hairpins. They use an accessory factor, the Rho protein P, to mediate transcription termination. Rho is a hexameric protein that hydrolyzes ATP in the presence of single-stranded RNA. The protein appears to bind to a stretch of 72 nucleotides in RNA, probably through recognition of a specific structural feature rather than a consensus sequence. Rho moves along the nascent RNA towards the transcription complex. There, it enables the RNA polymerase to terminate at Rho-dependent transcriptional terminators. Like independent terminators, these signals are recognized in the newly synthesized RNA rather than in the template DNA. Sometimes, the Rho-dependent terminators are hairpin structures which lack the subsequent stretch of U residues which are required for Rho-independent termination. B3. Transcription in eukaryotes. The mechanism of eukaryotic transcription is similar to that in prokaryotes. However, the large number of polypeptides associated with the eukaryotic transcription machinery makes it far more complex. 3.1 Eukaryotic RNA polymerases. 3 different RNA polymerase complexes are responsible for the transcription of different types of eukaryotic genes. The different RNA polymerases were identified by chromatographic purification of the enzymes in elution at different salt concentrations. Each RNA polymerase has a different sensitivity to the fungal toxin alpha-amidin, and this can be used to distinguish their activities. Institute for Advanced Studies, Jodhpur 18. RNA polymerase I. RNA pol I transcribes most rRNA genes. It is located in the nucleoli and is insensitive to alpha-amidin. RNA polymerase II. RNA pol II transcribes all protein-coding genes and some small nuclear RNA, snRNA genes. It is located in the nucleoplasm and is very sensitive to alpha-amidin. RNA polymerase III. RNA pol III transcribes the genes for tRNA, 5srRNA, U6snRNA and certain other small RNAs. It is located in the nucleoplasm and is moderately sensitive to alpha-amidin. In addition to these nuclear enzymes, eukaryotic cells contain additional polymerases in mitochondria and chloroplasts. RNA polymerase subunits. All 3 polymerases are large enzymes containing 12 or more subunits. The genes encoding the 2 largest subunits of each RNA polymerase have homology-related DNA coding sequences to each other. All of the 3 eukaryotic polymerases contain subunits which have homology to subunits with the E. coli core RNA polymerase alpha 2 BB. The length of each eukaryotic RNA polymerase is similar to the beta subunit of the E. coli polymerase, and the second largest subunit is similar to the beta subunit which contains the active site of the E. coli enzyme. The functional significance of this homology is supported by the observation that the second largest subunits of the eukaryotic RNA polymerases also contain the active sites. Subunits which are common to RNA pol I and RNA pol III, and a further subunit which is specific to RNA pol II, have homology to the E. coli RNA polymerase subunit. At least 5 other smaller subunits are common to the 3 different polymerases. Each polymerase also contains an additional 4 to 7 subunits which are only present in one type. RNA polymerase activity. Like bacterial RNA polymerases, each of the eukaryotic enzymes catalyzes transcription in a 5' to 3' direction and synthesizes RNA complementary to the antisense template strand. The requires the precursor nucleotides ATP, GTP, CTP and UTP and does not require a primer for transcription initiation. Eukaryotic RNA polymerases, unlike the purified bacterial enzymes, require the presence of additional initiation proteins before they are able to bind to promoters and initiate transcription. STD of RNA pol II. The carboxyl end of RNA pol II contains a stretch of 7 amino acids that is repeated 52 times in the mouse enzyme and 26 times in yeast. This heptapeptide has the sequence TRS or pro-THRS or pro-SR and is known as the carboxyl terminal. Domain or CD. These repeats are essential for viability. The CTD sequence may be phosphorylated at the serines and some tyrosines. In vitro studies have shown that the CTD is unphosphorylated at transcription initiation, but phosphorylation occurs during transcription elongation as the RNA polymerase leaves the promoter. Since RNA pol II catalyzes the synthesis of all of the eukaryotic protein-coding genes, it is the most important RNA polymerase for the study of differential gene expression. The CTD has been shown to be an important target for differential activation of transcription elongation. 3.2 RNA polymerase II genes, promoters and enhancers. RNA polymerase II, RNA pol II, is located in the nucleoplasm. It is responsible for the transcription of all protein-coding genes and some small nuclear RNA genes. Pre-mRNAs must be processed after synthesis by cap formation at the 5' end of the RNA and poly A addition at the 3' end, as well as removal of introns by splicing. Promoters. Many eukaryotic promoters contain a sequence called the tau box around 25 to 35 BP upstream from the start site of transcription Fig. 8. It has indeed consensus sequence 5' tata 8T-3, although it is now known that the protein which binds to the tau box, TBP, binds to an 8 BP sequence that includes an additional downstream base pair, whose identity is not important. The tau box acts in a similar way to an E. coli promoter -10 sequence to position the RNA pol II for correct transcription initiation. While the sequence around the tau box is critical, the sequence between the tau box and the transcription start site is not critical. However, the spacing between the tau box and the start site is important. Around 50% of the time, the start site of transcription is an adenine residue. Some eukaryotic genes contain an initiator element instead of a tau box. The initiator element is located around the transcription start site. Many initiator elements have AC at position -1 and an A at +1. 1. Other promoters have neither a ta-ta box nor an initiator element. These genes are generally transcribed at low rates, and initiation of transcription may occur at different start sites over a length of up to 200 bp. These genes often contain a GC-rich 20 to 50 bp region within the first 1 to 200 bp upstream from a start site. Upstream regulatory elements. The low activity of basal promoters is greatly increased by the presence of other elements located upstream of the promoter. These elements are found in many genes which vary widely in their levels of expression in different tissues. Two common examples are the SP1 box, which is found upstream of many genes both with and without ta-ta boxes, and the CCAAT box. Promoters may have one or both or multiple copies of these sequences. These which are often located within 1 to 200 bp upstream from the promoter are referred to as upstream regulatory elements, UREs, and play an important role in ensuring efficient transcription from the promoter. Institute for Advanced Studies, Jodhpur 19. Enhancers. Transcription from many eukaryotic promoters can be stimulated by control elements that are located many thousands of base pairs away from the transcription start site. This was first observed in the genome of the DNA virus SV40. A sequence of around 100 bp from SV40 DNA can significantly increase transcription from a basal promoter even when it is placed far upstream. Enhancer sequences are characteristically 10 to 200 bp long and contain multiple sequence elements which contribute to the total activity of the enhancer. They may be ubiquitous or cell type specific. Classically, enhancers have the following general characteristics. They exert strong activation of transcription of a linked gene from the correct start site. They activate transcription when placed in either orientation with respect to linked genes. They are able to function over long distances of more than 1 kilobit whether from an upstream or downstream position relative to the start site. They exert preferential stimulation of the closest of two tandem promoters. However, as more enhancers and promoters have been identified, it has been shown that the upstream promoter and enhancer motifs overlap physically and functionally. There seems to be a continuum between classic enhancer elements and those promoter elements which are orientation specific and must be placed close to the promoter to have an effect on transcriptional activity. 3.3 General transcription factors for RNA polymerase II basal transcription factors. A series of nuclear transcription factors have been identified, purified and cloned. These are required for basal transcription initiation from RNA pol II promoter sequences in vitro. These multi-subunit factors are named transcription factor IIA, 1 meter, etc. TFIIA, etc. They have been shown to assemble on basal promoters in a specific order, Fig. 9, and they may be subject to multiple levels of regulation. TFIID. In promoters containing a ta-ta box, the RNA pol II transcription factor TFIID is responsible for binding to this key promoter element. The binding of TFIID to the ta-ta box is the earliest stage in the formation of the RNA pol II transcription initiation complex. TFIID is a multiprotein complex in which only one polypeptide, ta-ta binding protein TBP, binds to the ta-ta box. The complex also contains other polypeptides known as TBP-associated factors TAF2S. It seems that in mammalian cells, TBP binds to the ta-ta box and is then joined by at least 8 TAF2S to form TFIID. TBP is present in all three eukaryotic transcription complexes, in SL1, TFIIB and TFIID, and clearly plays a major role in transcription initiation. TBP is a monomeric protein. All eukaryotic TBPs analyzed have very highly conserved C-terminal domains of 180 residues, and this conserved domain functions as well as the full-length protein in vivo transcription. The function of the less conserved N-terminal domain is therefore not fully understood. TBP has been shown to have a saddle structure with an overall dyad symmetry, but the two halves of the molecule are not identical. TP interacts with DNA in the minor groove so that the inside of the saddle binds to DNA at the ta-ta box and the outside surface of the protein is available for interactions with other protein factors. Binding of TBP deforms the DNA so that it is bent into the inside of the saddle and unwound. This results in a kink of about 450 between the first two and last two base pairs of the 8BP ta-ta element. TP with a mutation in its ta-ta box binding domain retains its function for transcription by RNA pol I and pol III, but it inhibits transcription initiation by RNA pol II. This indicates that the other two polymerases use TBP to initiate transcription, but the precise role of TBP in these complexes remains unclear. Institute for Advanced Studies, Jodhpur 20. TFIIA. TFIIA binds to TFIID and enhances TFIID binding to the ta-ta box, stabilizing the TFIID DNA complex. TFIIA is made up of at least 3 subunits. In vitro transcription studies, as TFIID is purified, the requirement for TFIIA is lost. In the intact cell, TFIIA appears to counteract the effects of inhibitory factors such as DR1 and DR2 with which TFIID is associated. It seems likely that TFIIA binding to TFIID prevents binding of these inhibitors and allows the assembly process to continue. TFIID and RNA polymerase binding. Once TFIID has bound to the DNA, another transcription factor, TFIIB, binds to TFIID. TFIIB can also bind to the RNA polymerase. This seems to be an important step in transcription initiation since TFIIB acts as a bridging factor allowing recruitment of the polymerase to the complex together with a further factor, TFIIF. After binding RNA polymerase. After RNA polymerase binding, 3 other transcription factors, TFIIE, TFIIH and TFIIJ, rapidly associate with the complex. These proteins are necessary for transcription in vitro and associate with the complex in a defined order. TFIIH is a large complex which is made up of at least 5 subunits. TFIIJ remains to be fully characterized. CD phosphorylation by TFIIH. TFIIH is a large multi-component protein complex which contains both kinase and helicase activity. Activation of TFIIH results in phosphorylation of the carboxyl terminal domain, CTD, of the RNA polymerase. Phosphorylation results in formation of a processive RNA polymerase complex and allows the RNA polymerase to leave the promoter region. TFIIH therefore seems to have a very important function in control of transcription elongation. Components of TFIIH are also important in DNA repair and in phosphorylation of the cyclin-dependent kinase complexes which regulate the cell cycle. Initiator transcription complex. Many RNA pol II promoters which do not contain a ta-ta box have an initiator element overlapping their start site. It seems that at these promoters TBP is recruited to the promoter by a further DNA binding protein which binds to the initiator element. TP then recruits the other transcription factors and RNA polymerase in a manner similar to that which occurs in ta-ta box promoters. B4. mRNA processing. There appears to be little or no processing of mRNA transcripts in prokaryotes. Ribosomes can assemble on and begin to translate mRNA molecules that have not yet been completely synthesized. Karyotic mRNA is degraded rapidly from the 5' end in the first cistron protein coding region, can therefore only be translated for a limited amount of time. Some cistrons are partially protected by stem loop structures that form at the 5' and 3' ends and provide a temporary barrier to exonucleases and can thus be translated more often before they are eventually degraded. Because eukaryotic RNA 2 transcribes such a wide variety of different genes, from the snRNA genes of 60 to 300 NT to the large antennapedia gene, whose transcript can be over 100 kilobits in length, the collection of products made by this enzyme is referred to as heterogeneous nuclear RNA (hnRNA). Those transcripts that will be processed to give mRNAs are called pre-mRNAs. Pre-mRNA molecules are processed to mature mRNA by 5' capping, 3' cleavage and polyadenylation, splicing and methylation. hnRNA synthesized by RNA-pol2 is mainly pre-mRNA and rapidly becomes covered in proteins to form heterogeneous nuclear ribonucleoprotein (hnRNP). The proteins involved have been classified as hnRNP proteins AU. There are two forms of each of the three more abundant hnRNP proteins, the A and C proteins. Purification of this material from nuclei gives a fairly homogeneous preparation of 30 to 405 particles called hnRNP particles. These particles are about 20 nanometers in diameter and each contains about 600 to 700 NT of RNA complexed with 3 copies of 3 different tetramers. These tetramers are A1, 3B2, A2, 3B1 and C1, 3C2. The hnRNP proteins are thought to help keep the hnRNA in a single-stranded form and to assist in the various RNA processing reactions. snRNP particles. RNA-pol2 also transcribes most snRNAs which complex with specific proteins to form snRNPs. These RNAs are rich in the base uracil and are thus denoted UL, U2, etc. The most abundant are those involved in pre-mRNA splicing UT, U2, U4, US and U6. However, the list of snRNAs is growing, and the majority seem to be involved in determining the sites of methylation of pre-RNA and are thus located in the nucleolus. Nucleoplasmic snRNPs are formed by the individual snRNAs complexing with a common set of 8 proteins, which are small and basic, and a variable number of snRNP-specific proteins. Proteins, known as the SM proteins, after an antibody which recognizes them, require the sequence 5' R8 U NGR3 to be present in a single-stranded region of the RNA. U6 does not have this sequence, but it is usually base paired to U4, which does. The snRNPs are formed as follows. They are synthesized in the nucleus by RNA-pol2 and have a normal 5' cap. They are exported to the cytoplasm where they associate with the common core proteins and with other specific proteins. Their 5' cap gains 2 methyl groups and they are then imported back into the nucleus where they function in splicing. 4.1. Capping 5' capping. Very soon after RNA-pol2 starts making a transcript, and before the RNA chain is more than 20 to 30 NT long, the 5' end is chemically modified by the addition of a 7-methylguanosine M7G residue Fig. 10. This 5'. Institute for Advanced Studies, Jodhpur 21. Modification called a cap and occurs by addition of a GMP nucleotide to the new RNA transcript in the reverse orientation compared with the normal 3' minus 5' linkage, giving a 5' minus 5' triphosphate bridge. The reaction is carried out by an enzyme called mRNA guanyl transferase and there can be subsequent methylations of the sugars on the first and second transcribed nucleotides, particularly in vertebrates. The structure forms a barrier to 5' exonucleases and thus stabilizes the transcript, but the cap is also important in other reactions undergone by pre-mRNA and mRNA, such as splicing, nuclear transport and translation. 4.2. Cleavage and polyadenylation at 3' end. In most RNAs, the mature 3' end of the molecule is generated by cleavage followed by the addition of a run, or tail, of a residues which is called the poly A tail. This feature has the purification of mRNA molecules from the other types of cellular RNAs, permitting the construction of cDNA libraries from which specific genes have been isolated and their functions analyzed. The cleavage and polyadenylation reaction requires that specific sequences be present in the DNA and its prem-RNA transcript. These consist of a 5' AAUAA3, the polyadenylation signal, followed by a 5' A3, where Y equals pyrimidine in the next 11 to 20 NT, Fig 11. Downstream, a GU-rich sequence is often present. Collectively, these sequence elements make up the requirements of a polyadenylation site. A number of specific protein factors recognize these sequence elements and bind to the pre-mRNA. One plex has assembled, cleavage takes place and then one of the factors, poly A polymerase PAP, adds up to 250 A residues to the 3' end of the cleaved prem-RNA. The poly pre-mRNA is thought to help stabilize the molecule since a poly A binding protein binds to it which should act to resist 3' exonuclease action. In addition, the poly A tail may help in the translation of the mature mRNA in the cytoplasm. Histone pre-mRNAs do not get polyadenylated, but are cleaved at a special sequence to generate their mature 3' ends. 4.3. Splicing. In the processing of pre-mRNA in eukaryotes, some sequences that are transcribed in which are upstream of the polyadenylation site are also eventually removed to create the mature mRNA. These sequences are cut out from central regions of the pre-mRNA and the outer portions joined. Intervening sequences, or introns, interrupt those sequences that will become adjacent regions in the mature mRNA, the exons which are usually the protein coding regions of the mRNA. The process of cutting the pre-mRNA to remove the introns and joining together of the exons is called splicing. Like the polyadenylation process, it takes place in the nucleus before the mature mRNA can be exported to the cytoplasm. Splicing also requires a set of specific sequences to be present, Fig 12. The 5' end of almost all introns has the sequence 5' GU3, and the 3' end is usually 5' AG3. The AG at the 3' end is preceded by a pyrimidine-rich sequence called the polypyrimidine tract. Up to is upstream of the polypyrimidine tract is a sequence called the branch point sequence which is 5' CURAY3, in vertebrates, where R equals purine and Y equals pyrimidine, but in yeast is a more specific sequence 5' UACUAAC3. Splicing has been shown to take place in a two-step reaction, Fig 12. First, bond in front of the G at the 5' end of the intron at the so-called 5' splice site is attacked by the 2' hydroxyl group of the A residue of the branch point sequence to create a tailed circular molecule called the lariat and free exon 1. In the second step, cleavage of the 3' splice site occurs after the G of the AG, as C2 exon sequences are joined together. Institute for Advanced Studies, Jodhpur 22. The intron is released in the lariat form and is eventually degraded. The splicing process is catalyzed by the U1, U2, U4, U5 and U6 snRNPs, as well as other splicing factors. RNA components of these snRNPs form base P pairs with the various conserved sequences of the 5' and 3' splice sites in E branch point, Fig 12. Early in splicing, the 5' end of the U1 snRNP binds on the 5' splice site and then U2 binds to the branch point. The tri-RP complex of U4, U5 and U6 can then bind, and in so doing the intron is looped R and the 5' and 3' exons are brought into close proximity.